This is the neuron going right to left. So on the right side, we have the synapse. Following, we have the axon. In the middle, we have the myelin sheath. And then we have the cell body. And in the middle of the left side, we have the nucleus. And at the top of the left side, we have the dendrite. This is DNA replication starting right to left. Everything will be explained. At the right, we have topoisomerase, which is just a ring. We have SSBP, which is just a little dot located on the top and bottom. Then we have DNA helicase breaking through. It's a triangle. And on the left, moving down, we have a rectangle, which is DNA polymerase. Then moving up beside the dots to the left of them, we have DNA primase. And to the left of that, it's a little isolated part. We have RNA primer. And then it is pointing at the left of the RNA primer. Those are Okazaki fragments. And to the left of the Okazaki fragments, we have a little piece, and that is DNA ligase. And finally, on the leftist side, we have another rectangle, which is DNA polymerase. This is the exocrine gland going top to bottom. So at the top, we have eyes. They secrete tears, mouth, saliva, skin, sweat, stomach, enzymes, breast, milk, liver, bile, pancreas, juices, and intestines, mucus. These are the endocrine glands. So going top to bottom. At the top, we have pineal, hypothalamus, pituitary, thyroid, parathyroid, thymus, adrenal, pancreas, for females, ovary, and for males, testes. This is for the ovaries going bottom to top. At the bottom, we have the vagina, then the cervix, then the endometrium, and then the little puffy things are the ovaries. And then we have the fallopian tube going up, and the uterus is at the top. This is for the penis, starting at the top, moving left, around, all the way back to the right. So at the top, we have the bladder, left urethra, left penis, that's at the tip. At the bottom, we have the testicles, the two balls. Then we have the scrotum. Moving up the right, we have the epididymis, the vas deferens, the urethra, the ejaculatory duct, the prostate gland, and the seminal vesicle. This is for chloroplast, moving right to left. So pointing at the empty space, we have a stroma, and then a stack of those rings is called a granum. A single ring is called a thylakoid. Pointing out on the outside, we have the outer membrane, and we also have an inner membrane, and then the line between the outer and inner membrane is called the intermembrane space. This is the mitochondrion moving left to right. So first we have the granules, which are the little ovals. We have the ribosomes next, pointing at the little dots, the cristae pointing at the cracks, the intermembrane space, just pointing on the inner side. We have the matrix pointing at around the middle. Then we move down, we have DNA, which is just pointing at the rings. We have the outer membrane pointing at the open space outer. And we have the inner membrane just pointing in the inner side. And up, we go back up, we have ATP synthase particles just pointing at those little circles. This is the nephron, starting left top to bottom, moving right top to bottom. So at the left, we have the proximal tubule, where HCO3, NaCl, H2O, and nutrients, and K plus are taken out, and H plus and NH3 are put back in. Then we have the Bowman's capsule, and we have the glomerulus. Then we have the descending loop of Henle and ascending loop of Henle. And at the descending loop of Henle, H2O is taken out, and at the ascending, NaCl is taken out. Then we move to the right side top, where we have the distal tubule, where NaCl, H2O, HCO3 are taken out, and K plus and H plus are put back in. And finally, we have the collecting duct, where urea and H2O are taken out. Now we have genetic mutations. We first have chromosomal mutations, which are any change in structure or number of chromosomes. So the first type we have is deletion, which is just removal, duplication, double, inversion, swapped. We have translocation. The first type of translocation is non-reciprocal, where parts are removed and inserted. And then we have reciprocal translocation, where parts are just swapped. Then we finally have insertion, where things are just inserted. And then we have genetic mutations, where only one gene is affected. We have point mutation, where one base is subbed for another. We have in-frame, where bases are added or deleted in threes. And we have frame shift, where one or more bases are added or deleted.